What characteristics of the brain cancer you are interested in?
Well, we're interested in understanding the heterogeneity in brain cancer. So brain cancer, in particular the adult brain cancer, they can be divided into many different subtypes based on the previous classifications designed by the World Health Organization. And there they've classified gliomas, in particular adult gliomas, into a variety of different grades and different histological subtypes. And these characteristics, or these molecular, histological characteristics, play an important role in clinical diagnostics and also types of treatments that are performed on the particular patients. And our focus and our interest is to tease this heterogeneity, but not looking at it from a pathological or histological point of view, but more from the molecular point of view, and looking at the molecular features of the tumor, and then using that as a way to understand the different subtypes that exist.
What's the difference between the histological view of the tumor and the molecular view?
Well, so for the past 100 years, the traditional way of understanding brain cancer, or actually dividing cancers into different groups, was basically looking at it under a microscope, or when the surgeon performs a surgery, the location of the tumor plays an important role, as well as the size and other features during the treatment. And this plays, so these characteristics kind of defines outcome, basically whether the patient will have a longer survival life, or whether they will have a particular treatment protocol, either a less aggressive treatment protocol or a more aggressive treatment protocol. And so what we are doing, with the advent of the newer technologies that have emerged in the last 10 years or 20 years or so, we are now able to tap into the genome, as we like to say, or the molecular features of these tumors. And what we are extracting from these tumors are DNA, RNA, and protein. And what we're doing is we're profiling a variety of different molecular features, such as transcriptomics, looking at the RNA or transcription level of the tumors. We're looking at methylation level, basically looking at CpG or DNA methylation level in the tumors. And we're also looking at proteomics and looking at the protein level. But then we're looking at whole genome sequencing, and we're looking at mutational landscape, the alterations that may be associated with that in the tumors. And given all that information, we try to understand whether the previous histological classifications or the other identifiers that were previously used, how do those relate with the molecular subtypings that we're coming up with? And that's one of the main motivation behind this study, is to kind of see whether these two fields come to an agreement, and if they don't, how can we use one or the other to improve on these classifications? Because at the end of the day, what is really important is to provide a proper treatment protocol or proper diagnostics for the patients, so that we can either learn how to extend their lives, or we can either tell the patient whether they'll have a better survival versus maybe a poor survival. And depending on these different outcomes, the clinician will then decide whether they would do a more aggressive treatment, like maybe do surgery and chemotherapy and other types of drug therapy, versus maybe doing more of a relaxed treatment, maybe it's just surgery or chemo. And this has an important aspect in the person's quality of life. So if we can not only improve the patient's survival, by maybe identifying these molecular marks that may help us understand these subtypes, that we can then identify possibly drug targets for these elements. But then the other thing is, can we also improve the quality of life of the patients? Because we don't want to give a patient an unnecessary treatment if they don't require that based on their molecular classifications.
Great. And just to make a first impression, do you think the histological classification and the molecular classification really get a consensus or not?
From our study, we see that there are some consensus in general, but by and large, we're seeing some inconsistencies. And this is kind of what we had almost expected, because there's a lot of intervariability between different pathologists. So if you were to get 10 neuropathologists together in a room and they all examine the same histological slide, almost 9 out of the 10, or I'm sorry, maybe 8 out of the 10 or so will come to an agreement. 20% may not come to an agreement, or it could be even higher, maybe 50 or 60%. And so this intervariability between diagnostics is an important key element that needs to come to a consensus here. And we feel like the molecular classifications that we're presenting in this new paper will help and aid clinicians, including the pathologists, to investigate the tumors, not only from a gross histological point of view, but also from a molecular classification, molecular profiling.
Is it going to replace the histological view?
I don't believe it'll replace it 100%. Because there's still some challenges to do a lot of the molecular thing. As I said, we're in a new era here in 2015, 2016, where technology and biotechnology has advanced considerably in the last 20 years. But we're still far away from being able to translate a lot of this really important biotechnology that we have at our disposal into the clinics. There are major efforts underway right now to translate these technologies into a more practical setting in the clinics, but it doesn't exist right now. I mean, it does exist, but it doesn't exist for the entire world. And so the new WHO that will be coming out in the coming months basically proposes that the first arm of classification should be done using molecular classification. But of course, if that does not exist for example, a developing country, then they should rely on the traditional classifications.
Great. Now let's get a little bit deeper in the paper itself. I'd like to explain first what's the Cancer Genome Atlas, and then we are going to take several aspects of it to improve the knowledge of your paper. So, great. What is the Cancer Genome Atlas?
So the Cancer Genome Atlas is a large consortium effort that was spearheaded by the National Institutes of Health in the United States. So the United States Science Institution basically decided that they needed to profile the molecular phenotypes of all these different tumors, the more aggressive ones and the more prominent ones in our society. And so what they did is they gathered together a variety of different institutions from all over the United States, anywhere from some USC, where I did my PhD, to Harvard and Broad and MIT in Boston, and a lot of different centers in between, where each of these different centers will provide a particular expertise in this particular study. So, well, let me try to think about this a little bit more.
Okay, no
I don't think I'm kind of going through it, and I'm thinking that maybe that's not probably the right way to say it.
But you can also, is it this one from here, right?
Yeah.
Oh, thank you. And you can also say that you have samples from all over. These other noises from your computer?
I don't know. I'm so used to the sound here. I'm not sure.
Yeah. Is it bad?
Is it bothering you?
It's okay here?
Assim, tem fundo, mas acho que não tá. Quando eu começo a falar, a voz tá bem estancada.
Okay. So you can
maybe
also
I can start again. I think
yeah,
better,
the cancer.
Yeah.
But you can be more objective than saying, for instance, it's important to have a large number of samples. It's important to have expertise from all the centers in the world and things like this.
Okay. So the Cancer Genome Atlas, or TCGA, basically started in early 2006 with the main effort and aims to characterize the molecular phenotypes of cancers. And then associate that with the clinical phenotypes and clinical outcomes. So with the ultimate goal of trying to identify candidate druggable targets, and then also to maybe improve treatment protocols for some of these very aggressive tumors such as brain cancer. And during the pilot phase of the TCGA project in 2006 up to 2009, the US government, which which started this project, which initiated from the National Institutes of Health, seeded about $500 million to get the project started with characterizing just 3 primary tumors: brain, lung cancer, and ovarian cancer. And with the success of these 3 primary tumor types, they then expanded the efforts for the additional 5 more years from 2009, and had just recently finished their efforts last year, December of last year of 2014, where they stopped collecting samples. But they have collected now up to 10,000 tumor samples from over 33 different tumor types that are very common among us. And so this effort really, the main motivation behind this effort was to gather as many samples as we can, but not only to get the samples, but to get a more of a heterogeneous pool sampling of the samples across a variety of different centers, so we're not focused on maybe one really high level hospital that may have the best protocol, but to get kind of a diversity in terms of treatment protocols from around the world. And in fact, we've been able to successfully expand our efforts from TCGA to gather samples from all over the world, including Korea, China, and here in Brazil as well. So several of our samples in this project have originated from here, from the Faculty of Medicine in Dr. Carlacci's tumor bank.
What about the idea of this research? Exactly what was the question, the first question you wanted to answer?
So this effort started in 2008, where we started to look at high-grade glioma. And that was the first brain tumor type that TCGA profiled. And from there, we've learned a lot. We understood that there are clearly differences in subtypes that we could not have ascertained by normal pathological review. And one of the subtypes that we've identified was this phenotype called GSIMP, or glioma CPG methylated phenotype, that we published in 2010, that defined a very subset of patients that were associated with this IDH1 mutation, and they were much younger at the time of diagnosis, and surprisingly they had a much better survival than all the other high-grade gliomas. Because high-grade glioma, grade 4 GBM or glioblastoma, has a median survival of 15 months, and it's a very poor, one of the most aggressive tumors next to lung cancer. And so to find a subset which represented about 10% of all GBMs to have a much better survival, almost 3 years more than all the other GBM, was astonishing to us. And to find that only with the DNA methylation and with the somatic alteration with this IDH1 mutation was kind of a novel idea. And so we published that in 2010. We've expanded that sample set from 290 to over 500 that we recently published in 2013. And at the same time, we've investigated the lower grade gliomas. These are the grade 2s, grade 3 gliomas, and that was a separate project from the grade 4 project. And what we found from there was also a very interesting subtype by somatic alterations of IDH1 and CODEL status and so forth. And we looked at all the different molecular phenotypes. And that was well characterized and published in New England Journal of Medicine just this past couple months. So this effort that we've done, that we've been working on for the past 2 years, was to really, now that we've got these 2 populations of CNS tumors, or adult brain tumors, with different grading, one with the high grade that has a really poor survival, and another one with the low grade but has better survival, but there are still some heterogeneous sample sets within them. We thought, well, if we put them together, could we learn something even more with the larger set? And that's what we set out to do. So we basically pulled in 1,122 adult gliomas from around the world, and we were able to profile methylation, RNA, proteomics on a subset of them. We were able to look at whole genome sequencing, basically looking at the genomic architecture of these tumors for a subset. We were able to also look at the exome sequencing, is to look at the mutations of each of the genes in the entire cohort, and copy number alterations. And so we were able to pool all of these resources together and try to see which of these molecular phenotypes defines glioma as a whole. And what we've come up with was a remarkable finding that that the samples divide out by IDH status, which is what we've already knew from prior. But given this large group of different grading, what we found is that there were low grades and high grades mixing together within these different subtypes, and that is something that we weren't really expecting to see, and that led us to investigate this heterogeneity issue. And what we then discovered was that independent of any of the other experimental protocols, whether you're looking at copy number expression, methylation, or genome sequencing, what we found is that the DNA methylation was able to define the subtypes much more definitively than any of the other experimental protocols. And each of these different subtypes by DNA methylation had a association with some clinical outcome. There were some really distinctive molecular features that define them, and this can cross many different areas, not only the clinical translational part, but also the tumor biology to understand the progression of this tumor in adult gliomas.
Great. We have to explain each of these subjects because people don't know what's IDH or anything like this.
Right.
So I took 1, 2, 3, 4, 5 of them and see. Just ask you if they are correct. The 5 we can explore these 5 ones.
Sure.
IDH 1 and 2. You have to explain that it's a genes and has some mutation that provokes something exactly.
Mm-hmm.
And IP in 19Q.
Q, co-deletion, right.
Yeah. And then telomere length, because people don't know it's exactly related to the survival of the cell. DNA methylation, and number of gene copies. So we are going to talk about each aspect individually now. So what's the first aspect you look at the cell to find?
So in our study, what we propose is that a patient that comes into the clinic should be defined by their IDH status, or the IDH1 or IDH2 mutation status. So if they have the IDH1 mutation status, they would then fall into that group, which represents about 46% of all gliomas. Then there's another group, the IDH wild type. Okay, the IDH1 gene, this mutation, it was described in 2008 by Bert Vogelstein. They were able to identify a subset of GBMs to harbor this mutation, which was kind of novel at the time. And IDH1 is a gene that's responsible for maintenance of the Krebs cycle or glucose metabolism in the mitochondria. So it's an important gene that converts, I think it's isoglucarate to alpha-ketoglutarate. 2-hydroxyglutarate. So let's go back.
It's an important gene that converts a molecular linked to energy that provides and do something. You don't have to.
Yeah, let me try to be more. Let me try to be more like more simple.
Please.
Okay.
Yeah.
So IDH1 mutation, IDH1 gene is an important gene in maintaining glucose metabolism in our cells. And what we find is that a subset of our patients, or a subset of glioma patients, harbor a mutation, a point mutation in this gene. And when these patients have this point mutation, the function of this gene now gets converted. A lot of times when people look at point mutations genes, generally the gene loses its function. But surprisingly, this particular gene gains a new function. And what it does is, instead of catabolizing the particular pathway in metabolism of glucose, it will then create a different metabolite. Is what we call alpha-ketoglutarate. And this particular compound, this metabolite, has been shown to inhibit certain enzymes that then leads to a very interesting pathway or phenotype, which is this DNA methylation phenotype. And so this association has been shown later on to be associated with each other.
And what is DNA methylation?
So DNA methylation is basically it's the modification on the DNA sequences. So as we know, we have 4 different nucleotides in our DNA: adenine, cytosine, thymidine, and guanine. And when we have cytosines and guanines paired together and string together in what we call an island, so there's a series of CPGs is what we call. These CPGs are generally associated within the promoter, or they're very near to a gene. So this then defines whether the gene would be expressed or not expressed. And what happens is that the C or the cytosine of the guanine becomes modified. So instead of changing the cytosine to a different nucleotide like we think of in genetics where we have a point mutation where a cytosine will get changed to a thymidine or vice versa. What's going on in DNA methylation is that we're seeing a modification to the cytosine. So the 5' carbon atom of the cytosine goes from a normal hydroxyl, which is a hydrogen atom, and that gets then methylated. It basically becomes a methyl group. And this methylation, what it provides is, it's an important functional process in normal development. It defines cellular phenotype, meaning that certain tissues in our body expresses certain genes at certain periods of time to define cellular phenotype. For example, the muscle cells versus the skin cells, they have different functions, they have different features or phenotypes. And those phenotypes are defined by which genes are being expressed. You know, which ones are turned on, which ones are turned off. And this regulation, what we call gene regulation of the genes, is mediated by epigenetics. And epigenetics, as I said, it's the modification of the DNA, such as DNA methylation, or we can also have histone modifications, and these are different modifications of the protein that lives with the DNA. And these proteins, if they get modified, also provides a context for whether the genes around them can be regulated or not.
So it's not a modification in the sequence of the nucleotides, it's another modification that changes the profile of the genome. I'd like to...
Exactly. Yeah, exactly. So the epigenetics and what it does is it modifies the DNA, but it doesn't alter the DNA sequences. Okay. So it modifies either the CPGs as I described, or it can modify, for example, the proteins that wrap around the DNA to hold the chromatin structure of the genome. And so these modifications have been shown to be associated with regulation of the gene. So it defines whether the gene will be turned on or whether the gene will be turned off. And so generally speaking, and this is not the case for all the time, or every single scenario, but if a gene's promoter or the CPGs near the gene are methylated, generally that would define that gene to be silenced. Not 100% of the time, but it mediates and has the potential to silence that particular gene.
So usually if you don't have this methylation in your cancer cells, because usually our cells don't have this profile of high methylation, is it? What I understand.
So what's important about epigenetics is that it plays a very important normal function in defining our cellular phenotype. So we have methylation in our cells in normal development, and even on our normal tissues. So as I said, we have about 14 to 20,000 genes in our human body. Not every one of these genes gets expressed in our skin cells. So only certain genes needs to be expressed to define our skin cell, like the color of our skin, you know, the profile of the skin. And those sets of genes that get turned on in the skin don't necessarily need to be turned on, for example, if you look at the stomach cell. The stomach cell has a completely different function. It's there to produce, you know, digestive enzymes, so that when you're eating, you're consuming your food, it can digest those. And so there, it doesn't necessarily need to express those particular genes that are expressed in the skin. So what determines the stomach cell's genes for those particular ones that are defined by the skin cell to be turned off is mediated by epigenetics. So in those tissues, if you look at the stomach cells, for those genes that are associated in the skin, you will see that they would be methylated. But in our skin, they wouldn't be methylated for those same genes. Okay?
What about the tumor cells specifically?
So what we see in tumors, which is kind of striking, is that when we look at the same gene in a normal counterpart, so what we're looking at is the matched normal. So we're not looking at, say, blood versus the tumor. We're looking at the tumor versus its matched normal. So essentially, if we're thinking of, say, brain, when we take the brain tumor out, we should have a normal representative tumor or tissue type that's not cancer, but it's brain. And so when we take those same two different tissues and now look at their epigenetics, what we see is like global loss of methylation, and generally global loss of methylation, but we see this hypermethylation within promoters. So what happens is certain genes that generally are hypomethylated in normal tissue become hypermethylated. So hypermethylation means gain of methylation, and hypomethylation means loss of methylation.
So they are more expressed in normal regular cells and then in...
Generally speaking, the normal genes that are necessary for the function of the brain, for certain genes they may be turned off in the cancer, and that's what we see. Yeah.
So so you said already two informations: the IDH and the DNA methylation. These informations are very important for your research, right?
That's right. Yeah.
So now let's talk about IP19Q.
Okay, so 1p19q represents the chromosome 1 P arm. So every chromosome has two arms, the P arm and the Q arm, or the small arm and the long arm. And what we see is in a subset of low-grade gliomas, there are a subset of these patients have a loss of the small arm of chromosome 1 and the large arm of chromosome 19. So these two co-deleted, and they happen together in the same patient. And these patients, surprisingly, live much better than all other low-grade gliomas. They have a better overall survival. They're more associated with older patients. But we don't really understand the function of 1p19q co-deletion. What we do know is that it's tightly associated with this clinical phenotype.
Do you know also that it has something to do with telomeres?
Yeah, so telomeres, yeah, but this is something we're talking about is the loss of a huge arm, like the complete arm of the 1p, which could be megabases. Yeah.
Okay, of course.
Yeah.
So let's talk about the how is it
telomeres?
pronounced? Telomeres, the
Yeah.
telomeres, and the third mutations or the out profile.
Okay. Well, so.
So let's start like the other feature we analyzed, or...
Okay. Well, yeah, so the other novel feature that we characterized in the study was looking at the telomere maintenance and certain genes that are associated with this deregulation of the maintenance of telomeres in brain cancer. And so telomeres are the ends of the chromosomes, and they usually have repetitive elements associated with them. And so during cell division and mitosis, as the cells begins to grow and divide, these telomeres can shorten over time. And this is generally, if you were to look at general aging, if you get an aging population versus a young population, and independent of cancer, you generally see an overall shortening of the telomeres. So these are the ends of the chromosomes. They just get smaller and smaller over time. And these are generally repetitive elements. And so these repetitive elements can help define, longevity, or sometimes they can be associated with certain, phenotypes. And this is also, these alterations have been associated with certain diseases, like a variety of different aging-associated diseases like Alzheimer's and Parkinson's. I mean, not 100%, but they've been associated with it. And they've also been shown to be associated with cancer. What we found in this study, what's already been reported in glioma, was that there are these particular genes that maintain the telomeres. So as I said, normally we lose telomeres, but we don't lose them very rapidly. We still maintain, if we lose some telomeres, we try to maintain it. And there are enzymes that maintain this. There are these telomerase, or these enzymes. And one of them is mediated by TERT, ATRX, other genes that are important in maintaining the telomere lengths as we get older and older. What we find is that in cancer, in particular in gliomas, the promoters of, say, for example, TERT, which is an enzyme, as I said, that helps maintain this telomere, this promoter has been shown to be mutated, and then this is associated with high expression of TERT, and this overexpression of TERT has been shown to be associated with gliomas and other cancers, but in particular in gliomas. as well as ATRX. And ATRX doesn't have a mutation in the promoter; it's just there's a mutation in one of its domains. But what we found, and what others have reported actually, is that these two mutations are mutually exclusive, meaning a patient will have either the TERT promoter mutation or the ATRX mutation. And this represents roughly about 80% or so of the gliomas that we have. But one of the novelty that we showed in our paper was that we were able to associate the TERT mutation and the ATRX mutation to telomere length. And this is kind of a novel finding. We were able to, given that we have whole genome sequencing data both in the tumor as well as in the blood for each of these patients, we were able to estimate the expected tumor, the telomere length of each of these patients in a normal, and then we compare that to the tumor, and what we show is that TERT promoter mutation, patients who have that have a significantly shorter telomere length, much more than expected, whereas the ATRX mutation has a higher, longer telomere length. So somehow this ATRX and the TERT, independent of each other, are going through two independent pathways and disrupting telomere maintenance.
And what does it influence the phenotype of the cancer?
Well, with this we don't really have a clear insight into yet, and this is something we need to investigate. But we don't really see a strong difference in survival for these patients. What we do see is that the younger patients generally have this TERT promoter mutation. They have overexpression of this TERT expression. And so basically what has been shown here is obviously the telomeres are deregulated, the lengthenings are being deregulated much more than what we expected from normal, and it's open now for investigation to explore how this can help us maybe target this for glioma as a whole, because it doesn't really associate with any long-term clinical outcome. It's more kind of associated with glioma as a whole, so maybe a target, because there are some drugs that are targeting telomere maintenance, and if we can understand the roles of TERT promoter and ATRX in gliomas and how they are mutually exclusive, and we can apply some of these other drugs that are used in other tumors like AML, leukemia, maybe this can help maybe guide some treatment protocols for gliomas.
Great. We have also
Is it bad?
Mas pega de inviabilizar de ouvir ele?
Não, tranquilo ouvir ele, mas
Mas aí tem, é na presa, não tem o que a gente fazer.
dá um trovão.
So let me see. Time not to get too late because they are expecting us as well. But we are going in a good. I think we have to go deeper because we are University of São Paulo, and I have to explain to people that don't know anything about science.
Yeah, for sure. No, this is great. I hope I'm clear. I'm not.
Yeah,
I don't want to go too kind of
much
specific.
Clear.
But okay.
Yeah. Now, can we continue? Yeah. I want you to talk about the copy of gene, the number of genes, and what does it mean in cancer cell. First of all, why this occurs? Why they have variation?
Right. So what happens in cancer is the genome becomes very unstable. We don't really understand what drives cancer. We don't know what is it a point mutation? Is it the deregulation of the expression of the genes? Is it methylation that's driving it? We have some evidences in other cancers that there might be some particular drivers, like BRCA1 mutation in some females, a subset of females that are inherited is tightly associated with breast cancer, and this could be a potential driver for that and so forth. But we don't really understand what really drives the cancer. What we do observe when we look at the genome,
we don't really understand what drives a regular cell to become a cancer
That's
cell.
right. So that's exactly right. So what we do not understand is what drives a normal cell that's functioning to become a cancer. We have some evidences that there might be a driver gene, or there might be specific DNA methylation changes or expression changes or copy number alterations that may play a role. What we do find when we look at the tumor of the molecular features of these tumor is that we see large instability of the genome. Basically, we see large gains of copy number or we see large losses of copy number
Copy
and-
number of what?
of genes or of chromosomes, large contigs of chromosomes. So as I said, we have a population of chromosomes in our genome, and we should have always 2 copies of each of our genes. This is normally how it should be. What we see in cancer is that sometimes we get multiple copies of these genes unnecessarily. So we maybe get 4 or 5.
So let's just wait for some.
Yeah,
So
we can.
sometimes in cancer we find
So, and sometimes in cancer we see large copy numbers of these genes, and sometimes we also see large losses. So obviously we only have 2 copies of every gene. So when we talk about loss, we're either saying loss of one of the copy of the genes or 2. So we say loss of 1 or loss of 2, and then we can have gains, maybe 3 or 4 or 16 or 20 copies of the same gene. And so if you have the same copy of the same gene, that might be associated with overexpression of that gene, and that might deregulate the cancer cell. But also the loss of these genes can also play an important role, because if these genes are important in maintaining the cell cycle pathway, if these genes are altered in some ways, that can lead to the normal cell into cancer. But again, as I said, we don't really understand what drives a normal cell to cancer. But what we do observe are these large copy number alterations.
Is this observation new from your study, or these are regular observations?
No, these are well-known observations. This is common in most cancers.
Why did you look at it then?
Well, so as I said, the study is, we didn't want to be focused on one particular molecular experiment, because we believe that capturing all of the molecular features will help us really inform better in the cancer or the tumor biology. So as I said, we looked at DNA methylation, gene expression, copy number alterations, and then mutation. And we looked at all of these together to see which of these features are more specific to brain cancer. And what we find is, there are some hallmark alterations in copy number. For example, we have the 1p19 code deletion, so these 2 arms that are deleted. We also have huge amplifications of certain chromosomes that is also associated with high-grade GBMs. But this is have always been described and it's known in glioma. And of course we looked at everything else, but what we found in this study is that the DNA methylation data provided a better resolution of the subtypes than any of these other markers that was identified prior. And so I think this is the novelty of this work.
Now, what were the exams performed to reach this data?
Exams?
For instance, you had a microarray of something.
Oh.
No, you don't have a micro. You just have an array of something. So I just want you to give a fast look at each exam. For instance, an array of RNA sees what genes are working on that cell.
Okay. Okay, so you mean like the experimental things that we did for this? Okay.
Yeah.
Okay, so to profile the molecular phenotypes of each of these cancers, we relied on biotechnology. And there's a lot of technologies out there that helps us to profile in large scale the gene expression. So we're looking at RNA levels or RNA transcripts. And this was done using RNA sequencing. So what we did is we were able to extract the RNA from each of these tumors and then do sequencing of all the RNA that's being expressed in the tumor. And then we can define which genes are being expressed, which ones are not. For DNA methylation, we used a platform, a microarray platform that profiles 450,000 CPGs in the entire genome. And this is just a snapshot of all the CPGs that currently exist in our genome. We have close to 20 million CPGs. But given the technology that we have, we were able to profile 450,000. And it seems to be enough for us to get a good snapshot of what's going on in the epigenomic profiling of gliomas. We also use next generation sequencing to look at the DNA sequences to see which genes are being mutated and then which positions of the chromosomes are being amplified or deleted by looking at copy numbers. And then we also extracted proteins. And for a subset of these patients, we were able to look and characterize the proteomics of the tumors. So, not only looking at the DNA, but we looked at the RNA, but we can also look now at the protein level and see whether the protein is deregulated in any fashion.
What are these proteins? Do you have anything to explain? For instance, you have this gene and makes this RNA, and the protein that results from it is a protein that involves in something, and it's different in glioblastoma, or do you have anything like
again,
an example?
again, in our study, what we see is that there are subtypes that are being defined, and these subtypes are associated with these outcomes. So, for example, one of the phenotypes that we found is this GSIMP low phenotype that have a loss of methylation that is being associated with genes that didn't turn on, and these genes are associated with cell cycle pathways. And what we are proposing is that the idea is that that these groups of patients, they have a poor survival, and it may be because they have their cell cycle pathways been deregulated, and that's one of the ongoing hypotheses that we have in this study here.
The cell cycle, you mean, is the when it's mature, when it divides, when...
Exactly. So a normal cell will go through different cell cycles, meaning that it'll copy its chromosome, it'll divide and grow, and each of these steps in the pathway are directed by certain genes that are responsible for whether to tell the cell whether it should grow, whether it should divide, and so forth. And generally what we find is that when a tumor is very aggressive, the cell cycle sometimes becomes deregulated and altered. And that's sometimes very associated with cancer. So as cancer grows, it's the same idea as the cell cycle kind of losing its trigger. It doesn't know when to stop growing. That's what the hallmark of cancer. And so this particular patient subset that we find in gliomas is that they are all the genes that are being altered by epigenetics and also RNA at the transcript level are all associated with cell cycle pathways. So we're seeing that these things are becoming deregulated in these patients, and because they have a poor survival, we are hypothesizing that the cell cycle pathways for these particular patients are deregulated, that may lead to this progression phenotype that we're observing.
So it's a worse scenario.
The worst scenario, yeah. Exactly.
I'm taking, because I classified myself all the groups.
Uh-huh.
So let's talk about the groups you are proposing.
Mm-hmm.
What is this new classification you're proposing?
So what we're proposing is an extension of the previous study that was published in New England Journal of Medicine that we were a part of, where we looked and said that gliomas should be divided by their IDH status first and foremost. So whether they have the IDH1 mutation gene or not, they would be classified into those two different groups. And then if they have the IDH1 status, then they would be divided into either CODEL status, meaning that they have the 1p19q deleted and non-deleted. And then if they're wild type, they would then have their own subclassification. What we find in this paper is that within the IDH1 mutation cohort, using DNA methylation, we can stratify the patients into three very distinct clusters or groups. And each of these groups has a very distinctive clinical outcome, clinical phenotype. So is that okay?
And so let's talk. First of all, please, if you can characterize if they have the IDH mutation or not. If they have the IDH mutation, probably they'll have a better prognosis or a worse prognosis. Now I'm really confused.
Yeah, right. So IDH, if they have IDH1 mutation status, they generally have a better prognosis than the ones that are IDH wild type. Okay, and that's what we show here in our study as well, and it's been shown in other studies as well. But what we see is that within the IDH1 mutation is that we see three very distinct subtypes defined by DNA methylation. So as I said to you before, IDH1 mutation confers a epigenetic phenotype, the CpG island methylated phenotype that we published in 2010. What we didn't know at that time was that there are subtypes of this, and now we can see that there are clearly three very distinctive subtypes, even though they all have the IDH1 status. Their methylation level for each of these groups is much different. One group has a much higher level of methylation, whereas the other one has a much lower level of methylation. And these subgroups are associated with clinical outcome. And then there are other facets of it which is also associated with this alterations in cell cycle pathways. And so the ones that have a lower methylation level that then overexpresses these cell cycle genes have a much poorer prognosis than the ones that don't have this phenotype. And so we are proposing that maybe the patients have progressed from this higher methylated state to this lower methylated state. And we showed this in our study using some paired matched sample sets that this may actually hold true. And so if this is true, then we have a progressive model that we can start to focus our research on and try to tease out this phenotype.
Does this happen in the same tumor or in another, when it comes back after a while?
So we have a very small cohort of these paired matched samples where we have the primary and then it recurred or relapsed, after 1 year or 5 years later. So we have a small cohort of them. We have about 12 pairs of these samples. And what we see is that the tumor comes back in the same place from where the surgery happened.
And the first time it was like that, and the second time like that?
Yeah, so the first time had a profile, maybe a very high methylation phenotype, and then when it came back, some of these patients shows a loss in methylation at these sites. So we suspect that maybe the patients that are in this low phenotype may have been a progressive state. If given enough time, they just may not have diagnosed early enough to catch it.
So it comes from a better outcome to a worse one.
Worse outcome, that's correct.
Can you speak? My voice won't enter.
Okay, yeah. So what happens is the GSIMP high or these high methylated phenotype may progress to this lower phenotype, meaning that they have a better overall survival at an initial higher methylated state, but then over time, maybe possibly they've lost their methylation, and then those patients then have a worse outcome.
Great. Let's talk about the group of IDH mutations and the 3 subgroups.
Okay.
Just to explain shortly what's the difference between them and the difference of clinical outcomes of clinical.
So the IDH1 mutation defines 3 very distinct subtypes. One this 1p19 code deletion status, as I mentioned earlier, and this group has a much better overall survival. There's another group, it's what we call the non-codel group, that doesn't have the 1p19 code deletion, but what it does have is a higher methylation phenotype, or this what we're calling GSIMP high phenotype, or what it stands for is glioma CpG island methylator phenotype. This one has an overall higher methylation level. It also has a very good overall survival, better survival outcome. And then the third group has a lower methylation phenotype compared to the GSIMP high, and we call this GSIMP low phenotype. And these patients do much worse in survival overall.
If we look at the whole group, we would conclude that the glioblastomas, that are the most aggressive one, should be in IDH mutation or in IDH no mutation, and why there's this escaper phenotype.
Right. So that's kind of going back to the old study that we did in 2008 and in 2010 when we studied the high grade GBMs, and what we found generally is that grade 4 GBM or glioblastoma is what you just described. More than 90% of them have an IDH1 wild type status. They do not have this mutation. There's a subset of the patients that do have this IDH1 mutation status that we define as being associated with this GSIMP phenotype, this hypermethylator phenotype that we published in 2010. And then what we see is when we combine the low grade and the high grades together, the subset of patients that have this IDH status no longer clusters with GBM, it clusters with the IDH mutants. And so now we have a mixed bag of both low grade and high grade gliomas that are defined by IDH1 status, and then we have another group of patients that are also mixed bags of low grade and high grade, but they're IDH wild type. And this is also shown in our paper, in the New England Journal of Medicine paper, where we looked at low grade gliomas, and about 8% of those gliomas have an IDH wild type phenotype, which is what we didn't expect to see. And so we believe that there's just another new subset of gliomas that has this low-grade phenotype, but has an IDH wild-type status. And so, because they don't have the IDH. So our study basically divides the data into two groups, IDH wild-type and IDH mutants. And then within the IDH wild-type group, it's a mix of both low-grade and high grades, and we see 4 distinctive clusters there by DNA methylation, 2 of which have already been well characterized before, this mesenchymal-like and classical-like. But what we see is that there's this other phenotype that wasn't reported before that has this really low methylation phenotype that's generally associated with pilocytic isocytomas, or this grade 1 gliomas, and these are generally associated with young individuals, pediatrics. We're looking at adolescents and pediatrics. But what we're seeing is that these are adults, so these are 20, 30-year-old patients that also exhibit a molecular phenotype similar to these pilocytic isocytomas. And so we believe that maybe we discovered maybe a new subtype of gliomas that wasn't characterized before. And these are enriched with certain somatic alterations and other features by RNA. But again, these were all defined by DNA methylation and not by any other experimental phenotypes.
And they have a better prognosis.
And surprisingly, they have the best prognosis. Right?
So what I want you to say, look with me, if you said that 10% of the glioblastomas are less aggressive than the other 90%, and 8% of the LGG have a more aggressive phenotype, I think you should compare, for instance, if we have only the pathologist to look at it, we will have a worse classification from 10% of the gliostomas
Right.
and 8% of the...
Possibly,
Yeah.
exactly. Yeah, exactly. So, generally the classifications by the pathologists, they get them right majority of the time based on our studies. But again, there are new subtypes that are emerging that if we can now use these molecular classifications in the clinic, we can then separate these patients out. And then some of these patients, if they, for example, the GSIMP low phenotype, because they have a worse prognosis, maybe you want to perform a much more aggressive treatment protocol for these patients as opposed to the rest of them. And then for the other side, there's a subset of IDH wild type patients that have a much better prognosis, and there you probably don't need to do the harsh treatment that generally people would do for those types of patients because they have that phenotype. And so there, we can improve, using these classifications, we can hopefully improve the quality of life for some of these patients by using these molecular classifications. If we can define them into these different novel groups, then the patients can decide then whether it's worthy for them to do this more aggressive treatment versus not doing more aggressive treatment.
You're being very clear.
Okay.
It's great.
Good.
I think we have almost everything. How did you conclude that the IDH is the best indicator?
So actually, what we did when we did the study, we did the study based on DNA methylation only. We didn't look at IDH status. We didn't look
Sorry, DNA methylation.
Yeah.
I spoke it.
It's okay.
incorrectly.
So we
How did you conclude that DNA methylation is the best way of looking and classifying cancer types?
Well, because as I said, the project allowed us to characterize all these different molecular phenotypes. So we actually investigated every single one of these experimental types: RNA, copy number, DNA sequencing, as well as DNA methylation. And all of the other experimental types, we weren't able to divide the patients as cleanly as we could with DNA methylation. So it was to our surprise that this had happened. It wasn't that we were focused on that. It just happened that it came to that conclusion. And so the DNA methylation provided, we believe, a better sensitivity to defining these groups into these different cohorts.
Is it important that you had data analysis here and you cross the data? I would like to explain this process.
Definitely. So the way that we did the analysis is a collaborative effort. And each, as I said, the TCGA project provides an opportunity for different expert people to come in to the group. We, as DNA methylation experts, came in and where we're recruited to do the DNA methylation analysis for the gliomas. Our collaborators from MD Anderson and from Columbia University, they then studied the RNA sequencing data, or the other ones studied the whole genome sequencing data, or the TERT mutation stuff. So each of them had their own expertise into this puzzle. And what we found as a group is that, whoa, the DNA methylation defines this group much better than any of the other experimental type, and this became the story for the paper.
And did it come from?
And it came
from?
And this and the entire effort for the DNA methylation came from here in our laboratory. Yeah.
So you said to me that it's important to have a specific biological formation to make a data analysis. I'd like to explain, why do you choose this profile among your students, for instance?
Why did I choose
biologists to make a data analysis?
Oh, okay. Well, I mean, so when we specifically when we talk about DNA methylation has this inherent biology to it, and it's not very trivial for a lot of people. So if you were to get someone who's a non-biologist to understand DNA methylation, it'll be a big challenge for them, and because of that, they won't be able to take the data analysis to the next level, which is this integrative analysis that we would like to do, integrating DNA methylation with gene expression, with copy number, and then to guide that analysis in that direction. And so what we believe is, a biologist needs to kind of drive the analysis here, so they have kind of an expert understanding of molecular biology, DNA methylation or epigenetics and gene regulation. And so as they begin to do their analysis, they can sit there, whether they're in the middle of the night, or whether they're at the beach doing their analysis, they can keep thinking about this problem, this biological question that they're observing, and they can then formulate in silico analysis to answer some of their questions that they may have.
So they're not just in silico analysis is not a regular term.
It's a kind of a new term within the last, say, 10 years as bioinformatics started to grow. Bioinformatics is the field of doing kind of analysis, computational analysis, but with the focus on the biology. Hence bioinformatics. So yeah, it's a field that's emerging, and then it allows people to ask questions and then to do analysis kind of in the real time. But then it's also working with large-scale data. Okay, and I think that's the main thing, is that bioinformatics has kind of emerged because of these growth of the data that's emerging now in the field in science, and so we need people with some expertise in data management and data structures and working with computers, but also people who have some strong background in statistics, so they can understand the statistical model that's being applied, but also the biology. And I think all three of these efforts, the computational part, the statistical part, and the biology, go hand in hand when we talk about bioinformatics. And so I'm a strong believer that bioinformatics is just another description of a molecular biologist, because it's the molecular biologist that's going to kind of really direct the analysis, the in silico analysis that we were going to be doing, or that you would do in a computer. So I believe that it's easier to train a biologist, the computational part and the statistical part, than to train a computational or a computer scientist, the biology part. That's more of a challenge in that direction as opposed to the other.
What do you expect? Yeah.
The air,
I think what do you expect after publishing the study? What do you expect to happen?
Well, I expect that the field of glioma. I mean, this is why there is this importance of using molecular classification.
It wasn't included in my list. I just thought about
because these
it
patients
right
who
now.
are IDH wild type, some of the things that we're working on is just pathology and said, okay, these are low-grade gliomas, trying to investigate the differences between those aggressive treatment on them. But in reality, what we've come up with in some of our analysis is that the changes that are happening are not happening in the normal fashion, meaning that the changes of loss of methylation are not generally happening at the promoters of the genes, but they're happening in these intergenic regions, and they're associated with very interesting functional elements of the DNA. And so we're actively kind of investigating this now as a follow-up to this this project. But my expectation is that this study will help expand not only people who are interested in the tumor biology, because there's a lot of biological questions that we try to answer with the study. It may help give some people some understanding of denamethylation and epigenetics in the progression of tumor. But we also hope that this will translate in the clinics, and that the markers that we've identified, because we were able to validate this in a completely independent data set, we believe that the markers that we've come up with by methylation are so robust that we hope that this could be applied in the clinic, and then the patients who come into the hospital will then be able to have these profiles done as a routine diagnostics, so that the patients and the clinicians can determine the best treatment protocol for the patient. And as I said to you before, the classifications prior to our work in the last 15 years or so have been very heterogeneous in terms of diagnosing these patients. And diagnosing a patient grade 2 or grade 3 can mean a big difference in the quality of life of the patient. It'll determine whether the patient should do an aggressive treatment or a less aggressive treatment. And you can get almost a flip of the coin with some of these pathologists in terms of grading. And so if we can use a molecular classification, a more objective way of defining these patients, it gives both the patients, their families, and the clinicians, the oncologists, more of a certainty of how to guide the patient treatment protocol. And I think that's where I would like to see this study go. I think that this would help. In long term, I think that if we would then hopefully find some drugs that can target some of these things, and maybe hopefully improve the longevity of these patients, to increase their longevity from 15 months to 24 months, or to 3 years or 5 years, so that they can see their grandchildren grow up. But we also want to improve the quality of life. We want them to be able to be coherent, and have a normal life, to be able to drive after their diagnosis, and not have to be so dependent on families and so forth for their disease. And so hopefully this study can alleviate the burden on the families.
Are these exams available?
So these exams are currently not available. We are working on making a panel, so that we can provide this as a clinical diagnostic panel for the clinicians. But it's a work in progress, and we hope maybe within the next 12 months we'll have something out there so that the people can use it. There are some really high-level institutions, advanced institutions in Germany and in Canada, who are using this array as a normal diagnostics protocol. It's kind of expensive. It's also challenging, because you would need a skilled people to analyze this type of data. So we're kind of far away from there. But our hope is that we can try to simplify this and make a very small panel that then can be used in the clinics very easily, routinely, as any other molecular classification.
Does all this knowledge applies to other tumors? To other tumors?
Yeah, so that's a good question. I think what we see is the denamethylation changes that we're observing in glioma is very distinctive for CNS or for glioma cancers. Other cancers like ovarian cancer, that is more defined by other molecular phenotypes, not by denamethylation. Colorectal cancer also can be defined by some denamethylation changes. Lung, for example, has other alterations that are associated with it. So every tumor type, every tissue type has their own defined molecular characteristics. It just so happens that gliomas, adult gliomas, have a very strong association with denamethylation and epigenetics.
But can you take any of this knowledge, this new knowledge, to another cancer research?
Yeah, and so TCGA is doing that now, and they published over 30 articles on all these different tumor types. And so the knowledges that we've learned from GBM and gliomas, the methods that we've developed here in our group, we've created a package, a bioinformatics tool that's now freely available online. And so people who have an expertise knowledge in, for example, bioconductor or in R can download our tool and reproduce all of our analysis freely. And of course, a lot of the analysis that we integrated in our paper are in this package. And so, yes, people can definitely use the knowledge that we've gained and apply it to any of the other tumor types. Definitely.
Sorry. What was exactly the contribution you and this group made to the project?
Yeah, so as I said, the TCGA project is a huge collaborative effort among many different institutions around the world, and we're very fortunate to be part of this consortium. And as I said, many institutions, either they provide their samples or they provide the disease experts, meaning a clinician that can come and help kind of understand the disease itself. And then there are data analysts or these analysis working group members, and they come in from different expertise. So as I said, people can come in from an RNA sequencing expertise, a whole genome sequencing expertise, or the epigenetics expertise. And so we were recruited into TCGA because of our experiences and skills with analyzing DNA methylation. And so that's what we provided for this consortium. What we've done, as I said, we've published now 4 other papers in GBM and gliomas, and now this is basically the last big marker paper that defines the CNS tumors for TCGA. And this is, and I've been very fortunate to be involved in the majority of these studies. So when we came together to work on this, there was no doubt in my mind that I wanted to get involved in this analysis. And so myself and Roel Verhaak and Antonio Alvaroni worked together leading this effort to understand glioma, both low grade and high grades, and to see how these heterogeneity differences that we can see both by the molecular classification and by histological classification, how that can come together. And so, yeah.
Can you say you're the man responsible, the group responsible for the conclusion of the project, for taking all the data and concluding these 6 or 7 groups classification?
That's right. That's right. So so our group basically led the effort in the epigenetics analysis, and we defined the 7 the 7 groups in GBM and gliomas. Actually, let's go back.
Yeah. No
I screwed that up.
Yeah.
So yes, our group was led the effort in identifying these very distinctive subgroups in gliomas, the 7 classifications defined by DNA methylation. And so that all originated here in our group.
Tão orgulhosa. And just to finish, is it important to have an interaction between clinicians and pathologists and researchers and this translational medicine that are being done here?
I strongly believe that's the most important way to really answer very important questions about diseases, in particular cancer. We need this crosstalk between clinicians and pathologists and molecular biologists, because the molecular biologists, if they're not guided properly and what the clinicians are seeing with the patients, we could be going in the wrong path. And as well, the clinicians need to also work with the molecular biologists, because as we provide new insights and new understanding of the tumor biology and the tumor genome, as we're looking at right now, we can help inform the clinicians so that they can have a better relationship with their patient and provide hopefully better treatment protocols, and quality of life for the patient. So I really believe that what we've done and what we can do and what people should do in general is to have this very nice close collaborations, this translational collaboration that a lot of people talk about, from going from the wet lab to the bench.
And the patent you said, is it about what?
So we're working to create a panel of probes that can define these groups that I previously described, and so we want to be able to characterize them a little bit more and to restore or retain the intellectual knowledge that we've discovered from these probes. And my expectation is that I'm not going to get any money off of this, but we would like to just patent it so that we can have the University of Sao Paulo's name attached to these important clinical markers that may go out into the clinics around the world.